the technology today, low productivity and stability issues can probably be targeted in one of two ways. One, you screen more clones, and sometimes that is necessary. You screen more clones, it's quite labor intensive, and that's the way you go. But at least if you're using random integration, you're finding that needle in a haystack, you're more likely to if you screen more. Or, as I mentioned earlier, you look at those semi-targeted integration technologies like transposons.
Welcome back to part 2 of our deep dive into cell line development with Andrea Gough from Advanced Instruments. If you missed part 1, make sure to check it out. In this episode, we'll explore the exciting intersection of artificial intelligence and cell line development, discover essential tips for newcomers to the field, and uncover systematic approaches to troubleshooting common challenges. Let's jump right back into our conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Let's talk about another aspect of cell line development for a minute, because as you've mentioned already, you're screening a lot of clones and you can get a lot of data really quickly, and it can become quite complex just to understand which is the best producing clone. And then if you include, for instance, product quality attributes, it gets even more complicated. So with the rise of AI and machine learning now in biotech, how do you see these technologies transforming clone selection and cell line optimization?
Yeah, AI is everywhere at the moment, and so it's inevitable for sure, but we're certainly using it, and I think people would be amiss if they didn't think of how could I possibly use this to help me. And a recent conference that was the topic of discussion in general. I would say whether you're a provider, i.e. like a service provider, or you're a developer, AI is going to get introduced everywhere. We're specifically using it to kind of take the workload off of the customer. So in that initial step where our instrumentation, the VIPs dispenses the cells, actually uses AI to say, is that a cell or not? And say, yes, you've got 1 cell, 2 cell, on to the next stage or next well. And then when the other thing we have is part of the process in CLD is really eyeballing every image of a clone and saying, did that come from a single cell? Well, we've introduced it in by assisting that process. So immediately the AI looks at the whole well and it immediately identifies the object, i.e. the cell that it's detected. And we've spent a lot of time feeding in images, thousands and thousands of images into our databases, our machine learning that enables our instruments to do that for customers. So the point of that is really to drive efficiency. And I think that I've heard some customers saying they want to use images and AI to help them see if there's anything that AI can pick up in the images that will indicate a good clone, whether that's stability or production or something along those lines, if they can do that earlier when you have a collection of images. So yeah, I think it's inevitable. And like I say, we're using it. And I know that there are people that are going to use it to try and almost predict the best clone. So I think that's the way forward.
And how will these technologies help us overcome certain expression limits, for instance, for difficult to express proteins?
That is a good question. I'm not sure I know. I think that's a difficult one because I think you would have to use AI and probably look on a closer scale of the cell, like go higher objective, maybe that would help. But for me, I'm not 100% sure on AI and how it could. I think it can and it probably will, but I'm more than sure that cells give off some form of phenotypic identifier. Some people in the past have thought if the cell's bigger, it's a good producer. So maybe there's things along those lines that can be used to help.
And what about other expression systems? I think we're still very much focused on CHO, especially in the biopharma world. Outside, I think there is a lot more innovation. Do you see any trends there?
Yeah, actually, CHO is holding fast when it comes to the protein therapy world. It's well known, and I think that people have a lot of success with it. It's highly robust. But when it comes to, say, viral vector production for gene therapy, we're definitely seeing a bit more of a mixed bag. HEC is definitely the most predominantly used cell line for AAV production, for instance. I know that there are other lentiviruses that are being produced with it. Generally, HEC is the most common we see. Sticking to that space, we have seen some customers that use HeLa, which I thought was interesting. So instead of HEC for viral vector production, people only tell us so much, of course, their proprietary information, so I can't delve too much, even though I'm interested. And then the other one I think we're seeing is MSCs, mesenchymal stem cells, for production of exosomes, and that's also another gene therapy delivery mechanism. There's definitely pros with exosomes in that space, so I think that that's going to get more prevalent for sure. And then the final one that we see a lot more of specifically as a solution provider is iPSCs, the use of iPSCs in allogeneic cell therapies. We definitely see that more, people wanting to move away from autologous cell therapies because highly expensive, it's personalized medicine, essentially. So there's some great work going on in that field, and I think that's just going to continue to grow. Oh, and I didn't mention insect cells when it comes to gene therapy. We also see a greater prevalence of insect cells. They have their pros as well over, say, HEC.
Andrea, you're talking to a lot of users, so I'm curious, what are the most common aspects of CLD that your customers ask for advice on in general?
I think every question you've asked so far is very good. Is hints and tips, to be honest. We get a lot of questions around, like, I'm not seeing outgrowth. Why is that? And we end up saying, well, it could be for a number of reasons. What are you doing before? What are you doing afterwards? What are you dispensing into, et cetera, et cetera. Regulatory, that comes up a lot. So I think questions like, are you FDA compliant? And it's kind of a really general statement. And I kind of feel like, in what respect are you asking? When we speak to customers, the more we probe, the more we can support. But yeah, I would say the most common is really regulator guidance and hints and tips for like the early stage of CLD. We also get some questions on how to automate, how to validate their processes. So we can often give hints and tips around that just from our experience of what we've gleaned over the years.
Let's make this now very practical. For scientists starting their first cell line development project, what 3 key pieces of advice would you give them to set themselves up for success?
I think based on my understanding, I would say really look for providers that can support you as much as possible with genuine experience from the field, whether that's a CMO or a provider like ourselves. I'd say look for technologies that are going to be flexible, modular, that can really suit your workflow, because even though everyone's doing very similar things, no one is doing exactly the same. I've had experience where there's 2 CLD labs in the same company using the same cell line, but they can't use the same media and they can't use the same plates because there's some weird difference between the 2. They just accept that fact. So I think that the workflow really should be as flexible as possible and make sure it's obviously compliant with their needs and requirements rather than like trying to get one size fits all, another solution out there. And then the biggest thing I would say, and we get this a lot, is like evidence. Like from the start to the finish, track everything, document everything. And then should you be questioned at a later date by the regulators, you know you've got it saved somewhere. I think it will really help CLD companies or developers if they've got everything they need up front. So even trivial things like certificates of analysis, people always forget about these and come back afterwards. What media has come in contact with your cells, what supplements, etc., etc. All of that information you kind of need in a dossier. All of your images, save your images, the raw images, the reports, the tracking. I would say that's the biggest thing for me. Those 3 tips is get someone that can support you, get flexible technology that's going to meet your needs, and then just track everything that you can up front rather than after the fact.
And even if you start right, you might encounter some low productivity issues or some instability issues. Are there any systematic approaches you recommend to troubleshoot these challenges?
I think the technology today, low productivity and stability issues can probably be targeted in one of 2 ways. One, you screen more clones, and sometimes that is necessary. You screen more clones, it's quite labor intensive, and that's the way you go. But at least if you're using random integration, you're finding that needle in a haystack, you're more likely to if you screen more. Or, as I mentioned earlier, you look at those semi-targeted integration technologies like transposons that, generally speaking, are said to give you higher levels of expression because of the multiple copy numbers that are integrated. And then the stability is very much related to the fact that the integration is targeted to a certain position. So I would say that they're the 2 aspects. Either if you can't go down the route of transposon technology, there are the companies out there that help people with that. I would set up processes that enable you to screen more clones faster and track them better, and also screen them along the way efficiently.
Fantastic. Thank you so much, Andrea, for sharing these precious insights. With all what we've covered today, what is the most important takeaway you want our listeners to walk away with?
I would say, from my learnings in general, the take-homes from our conversations and the conversations I have with developers is everyone's striving to do it as fast as possible, which makes total sense. The drug development process is really a long process, and we'll all benefit from it eventually. We all get ill. But I think making life easier for themselves in early stages is the take home for me. Get the processes set up front, get the systems in place, get support with that, and really get the documentation. Even if you don't ever need it, it's better than having to try and find it at a later date should you be questioned on something. So that would be my take home, is really pull together everything you need up front, get the right systems and processes in place, and then hopefully that will enable the CLD process to be as easy, as efficient as possible.
Andrea, where can people connect with you?
So I'm on LinkedIn. You'll see me on LinkedIn. The fastest way is probably just to message me via that. I have an email address. It's Andrea Gough at aicompanies.com. If anyone wants to email me, I'm more than happy to be emailed. But yeah, LinkedIn is probably the fastest way.
Great. I'll leave the link down in the show notes, so please do reach out to Andrea. And Andrea, thank you so much for sharing these great insights and for being on the show today.
Great. Thank you very much for having me.
And there you have it, smart scientists. A massive thank you to Andrea Gough for sharing her invaluable insights into the future of cell line development and practical tips for success in this field. And I thank you for tuning in. And could you just hit pause for a second and leave us a review? It will mean the world to me. And before we go, if you're looking to optimize your biologics development process, remember, you can develop biologics better, faster, at a fraction of the cost with our fractional CTO services. If this is of interest, book a free call with me with the link in the show note. Until next time, keep innovating and making bioprocessing smarter. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you.
For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.